Hi everyone, I'm Jason from Team A. I'm a student at the University of Illinois and I study molecular and cellular biology. Today, I will be discussing what I did as an assistant in the Tumor Research Lab during the past few weeks. First, I will talk about some techniques that this lab has been using, and then I'll explain what questions they're trying to answer and how these techniques work in application. A research lab cannot put its raw imported materials directly into experimentation. Preparation work must always be done so that all factors in the experiments are controlled and calculated. In this video, Shizang starts with a petri dish with liquid FBS medium that has been culturing tumor cells that they ordered. FBS medium, or fetal bovine serum, is filled with nutrition for cells to feed off of. Tumor cells are adherent, so they have stuck onto the bottom of the petri dish throughout their growth. Therefore, Shizang can suck the petri dish dry of FBS medium without disturbing the tumor cells. As you can see, he contacts the side of the dish with the sucker to avoid sucking off the tumor cells. After sucking off the FBS, he rinses the tumor cells with phosphate-buffered saline. PBS will rinse off any junk like random ions or serum, so that we're left with just the tumor cells, but it doesn't detach the tumor cells from the bottom of the dish. He then adds trypsin to detach the cells from the dish. Shizang then determines the concentration of tumor cells in his solution using hemocytometer cell counting. In this technique, cells are displayed on a grid like this. Note that this grid has 4 quadrants. He will count cells in these quadrants and disregard the rest. This becomes important later. To start, 1 milliliter of the tumor solution is mixed with 1 milliliter of this fancy blue dye. This blue dye, called trypan blue, will not penetrate the membranes of living cells. However, it will permeate through dead cell membranes and mark them as blue so that Shizang knows not to count them. Shizang then utilizes this incredible finger action to count the living cells that he sees. After counting all the cells, he uses some mathematics to determine the concentration of cells in his 1 milliliter of tumor cell solution. He originally counted 200 living tumor cells among the 4 quadrants, but this method of counting cells treats each of the quadrants as its own sort of random sample. Therefore, the first step in his math is to average the cells per quadrant. He then multiplies the expression by 2 to account for the 1 milliliter of trypan blue dye added to the 1 milliliter of tumor solution. Finally, he multiplies the expression by 10 to the 4th power. This factor accounts for the smaller volume of solution in each quadrant. Now that he knows the tumor cell concentration in his solution, he can determine how much volume of FBS he needs to dilute his solution to a desired concentration of tumor cells that is suitable for treatment. Now that we've treated the tumor cells with different silencing RNA, we will collect the RNA from the tumor cells to see how much gene knockdown has occurred. In this setup here, we first add trizol to an Eppendorf of tumor cells. Trizol completely lyses cell membranes and cell structure while inhibiting RNA breakdown. Next, chloroform is added to separate RNA, DNA, and proteins into separate layers. Centrifuging the Eppendorfs also help with this process. After this step, RNA will be suspended in the topmost layer. This RNA layer is then sucked off and contained into a second Eppendorf. The original Eppendorf with the remaining DNA and proteins is discarded. Next, isopropanol is added to the suspended RNA. This precipitates the RNA so that it clumps into a small RNA pellet. Centrifuging can also help with this step. Finally, all of the liquid non-RNA junk is sucked off, and the RNA is washed with ethanol because ethanol evaporates quickly. After the RNA dries off, the RNA is then resuspended in water. The next step is to create cDNA or complementary DNA through reverse transcription of the RNA. Essentially, the RNA is mixed with nucleotides and primers. Specific primers encourage the synthesis of specific genes. Earlier in this experiment, we treated the 2 samples of tumor cells with 2 different siRNA. However, we now split these 2 groups even further into 4 total groups by using 2 different primers. These 2 primers are for the DLG5 and GAPDH genes. Once again, the DLG5 primer aids us in reaching an experimental measurement on DLG5 expression after gene knockdown, while the GAPDH primer is only used as a control. After the cDNA is formed, we use qPCR to amplify the cDNA. This is done in a fancy, expensive machine. This will amplify our cDNA to give us a clearer idea of how much RNA of each gene type remains after RNA silencing. In order to analyze the results from the qPCR machine, one must first understand how qPCR works. In one cycle of qPCR, each double-stranded DNA is denatured, primed with primers, and those primers are extended with DNA polymerase. This newly synthesized DNA is then marked with cyber green and recorded by the machine. The machine will then record the number of cycles that it takes for the cyber green fluorescence magnitude to reach a certain threshold. So if we started with a smaller amount of DNA, it will take a larger number of cycles to reach that threshold, and vice versa. Here is the data that the machine gave us. Don't look at this column, it's trash. I wonder who did that column. This data is very good. 一定是. Never mind. CQ means the number of cycles until the threshold was reached. This box here gives details on the siRNA and the primer used in each row. The first step in the analysis is to subtract the GAPDH CQ from the DLG5 CQ for each sample. This solves for a value called the DLG5 delta CT. We then average the DLG5 delta CT in SIGFP values. We only use the SIGFP values to average because that is our control group that we will compare the SIDLG5 group to. Then we find the difference between the mean DLG5 delta CT in SIGFP with each DLG5 delta CT value. Finally, we take the negative log base 2 of all of these values because it gives the values some more meaning, and we're left with ratios of RNA expression levels comparing the SIDLG5 group to the SIGFP group. While this SIGFP group will still express 0.991 amount of RNA, this SIDLG5 group will only express 0.247 amount of RNA. Well, we just learned that our SIDLG5 RNA is silencing the DLG5 gene pretty well. But what does that really mean? What even is DLG5, and how can doing more research on it help us fight cancer better? Disc's large homolog 5, or DLG5, regulates cell growth and proliferation. It interacts with many different signaling molecules, which often lead to uncontrolled cell growth. But the truth is, we don't know too much about it yet. Dr. Liu Shen's lab is currently working on understanding the more complex unknowns of tumor cell signaling, to put it briefly. Labs like hers are important even just to gather more experimental data to add to bioinformatics databases. Thanks to research of the past, researchers of the future have data to base their experiments on, pharmacists have information to make treatments from, and patients can become more and more confident in modern healthcare. Special thanks to Dr. Liu Shen for letting me assist in her lab in the past few weeks.